This is a revision video on sliding filament mechanism of skeletal muscle contraction. So a lot of students struggle with this process, so go through this carefully if you are struggling to understand this. Repeat it as many times as you need to in order to get questions correct. So there are 6 questions. Pause the video, attempt the questions, and then we will go through answers. So question 1, what is a sarcomere? You should have this answer. Okay, and let's move on to question 2 then. So draw and label a sarcomere. So let's go through this step by step. So you should have drawn myosin, also known as the thick filament, so it appears thicker. And then actin, also known as the thin filament. And then you should have had some other labels here. So you've got the Z lines, the space or the structures between the Z lines is the sarcomere. You've got the M line, that central line of the myosin. And then I'm going to add some extra reference points here just so you can see what we're referring to. So the ends of the actin on both sides, that is the H zone. So I'm just helping you with those dotted purple lines to see exactly where they are. So I'm going to take them away now because we're going to use some others here. So the ends of the myosin, that is the A band. And then from the ends of the myosin to the Z line is the I band. Now, in reality, we don't see sarcomeres just individually like this, although it's helpful to label them. So I'm just going to add some extra information here, which is the other parts of sarcomeres connected to that sarcomere. But remember, a myofibril is going to have many of these sarcomeres. So in fact, this then allows us to also consider this I band, and so you can draw the I band as being this part, so extending not just to the Z line, but to the ends of the adjacent myosin. So that's also the I band too. So it depends on what you're shown if you are given a diagram, but it's good to be aware of that. So there is the basic structure of a sarcomere. So what happens when it contracts? How does it change? Let's take a look at that. There we go, animation shown. So let's have a look at what's changed here. So the I band has become narrower or shorter. The H zone has also become narrower and shorter. And the sarcomere itself has become narrower or shorter, and those Z lines are closer together. So you should have those things have changed, become shorter or closer together. The things that haven't changed are the position of the M line and the length of the A band. Those have remained the same. Okay, so just a few things I wanted to mention before we move on to the next part. So it's worth remembering that in reality, there are many sarcomeres connected together, and many sarcomeres together are going to form a myofibril. So many layers of sarcomeres, and amongst those sarcomeres, it's worth remembering that you are going to find mitochondria, which are supplying the ATP for the process of muscle contraction. So it's just remembering that, well, worth remembering that structural feature of muscle. So I'm just going to switch back to that basic diagram of a few sarcomeres again. To this diagram here, and you'll notice that all I've done is change the colour. Now I've done that just simply because for the remainder of the animations and diagrams, I'm going to depict myosin as yellow and actin as blue. So I'm just trying to help us understand this process by having colours here. So what we're going to do is we're going to focus on a region where I've put the black box here. So we're going to focus in on that part. And so when we focus in, this is what we'd see. So in blue, we've got the actin, and in yellow or orange here, we've got the myosin, and you can see the myosin tail, lots of tails connected, and then lots of myosin heads protruding from those myosin tails. And so again, we're going to focus in even more before we get into this process of the sliding filament theory. So I'm going to draw a box here, and we're going to focus on that part. So here we are. We focus right in now, and what I wanted you to do was to tell me 3 main proteins of the sarcomere. So you should have actin, and describe that is a globular protein, but you've got many actins bonded together to form the whole actin molecule. You've got this red structure here, tropomyosin, and you should have also identified myosin as being a key protein. And remember that myosin has a myosin head, which is also an ATP hydrolase or ATPase, and it has a tail, so this fibrous protein tail and this globular head. So myosin has 2 parts. Okay, so let's get into the process of sliding filament mechanism. So I wanted you to put this into 6 steps. So we're going to go through this carefully and have an animation, so hopefully that helps you see this process and how it all connects together. Okay, so we're looking at 3 myosin heads here next to the actin, and we've got the tropomyosin in red. So let's go through this process. So first thing you should have mentioned in your steps is remember that we've got ADP and PI bound to those myosin heads. But the first thing you should have mentioned is this is about to be animated. And so we've got calcium released from the sarcoplasmic reticulum that increases calcium in the sarcoplasm. Those calcium ions bind to the tropomyosin, and they cause it to change its tertiary structure or change shape, and that reveals the myosin head binding sites on actin. So I've shown those myosin head binding sites as sort of paler blue on the actin there. So those circles, those lower circles. Okay, next step. So you should have written myosin head can now bind to actin, forming this actinomyosin bridge. Now what you'll have noticed with this diagram is I've numbered the individual actin proteins in the whole actin structure. Now I've done that just to help you follow how that actin moves along. So hopefully we'll go through this process. We're going to go through this process twice, so you'll see how myosin pulls the actin along by referring to those numbers. So you can see here that the myosin head is bound to number 1 actin, number 6 actin, and number 10 actin. So let's go on to the next step, step 3. So you can see that ADP and PI leaves the myosin head. And next step. And so that ADP and PI leaving caused that myosin head to change shape, and it swings forward, pulling the actin along. That is the power stroke. Okay, so next step. So ATP then binds to the head, causing it to detach from the actin, breaking that actinomyosin bridge. There's a key role of ATP there. Okay, and final step. Okay, so there are a few things going on there. So you should have written this down. So remember the myosin head is an ATPase or ATP hydrolase, and that is calcium dependent. So that hydrolyzes the ATP, releasing energy for the myosin head to change shape, to move back to that starting position, to that original position. Okay, so what we'll do is we'll go through that process again. So you'll see that myosin is now going to bind to a different part of the actin. It's pulled the actin along, and you'll see how it binds again to further along and then pulls the actin along again. So let's just go through that process. I'm not going to add in the... Oh, I'm going to add in the descriptions, but I'm not going to number them this time. But let's just do that again, step by step. So you can see here that now, because the actin's been pulled along, the myosin head now binds again, but at number 2, 7, and 11 of the actin molecule. And so let's keep going. So that actinomyosin bridge is broken because ATP binds to the myosin head. And that final step, hydrolysis of ATP to ADP and PI by the myosin head, which is also an ATP hydrolase, ATPase, and it returns to that original position. Now, what if the stimulation from the motor neuron is no longer present? What happens then? How does this muscle contraction stop? So you should, for this, have written this. So if that stimulation from the motor neuron is no longer there, then what happens? I'll show you in the animation. So the calcium ions are actively transported back into the sarcoplasmic reticulum. That means that they are no longer bound to tropomyosin. Tropomyosin changes shape, and it covers those myosin head binding sites on actin again. So we can't get an actinomyosin cross bridge forming. Okay, so the last thing was to summarize the role of ATP and calcium ions in this process. Let's take a look at the answers for that. So for the role of ATP, you should have written 3 things. So it binds that myosin head, causing it to detach, breaking the actinomyosin bridge. It's hydrolyzed by the myosin head, which is an ATP hydrolase or ATPase, and that releases energy to cause it to move back to its original position. And when there is no longer stimulation from the motor neuron, then its ATP is hydrolyzed to release energy for active transport of calcium ions back into the sarcoplasmic reticulum. It's really important that when you phrase that, you don't write ATP is used to actively transport calcium ions back into the sarcoplasmic reticulum. It's really important you be careful with phrasing there. It's hydrolyzed to release energy for active transport. And finally, calcium ions, what is their role? So 2 key roles here. Binds tropomyosin, that causes it to change tertiary structure or shape. to allow that myosin head to bind to actin. And remember, the ATP hydrolase on the myosin head is calcium dependent. Okay, so I hope that was a good summary and that helped you understand the process of sliding filament theory of muscle contraction. If you're still not quite getting it, then have another go at the questions, try again until you are getting all the questions correct.